Welcome everyone to our Wednesday seminar. It's my pleasure to introduce Henrique Carvalho, who is an assistant professor at the University of Manitoba in the Department of Soil Science. Henrique received his MS and PhD at Texas A&M University. He specializes in micrometeorological techniques, looking at trace gas, mass, and energy transport between the land surface and atmosphere. And his seminar today is Blowin in the Wind, Micromet Research in the Black Soil Zone of the Canadian Prairies. So thank you, Henrique, for doing this.
Thank you, Tim, for the introduction and also for the invitation. Yeah, sorry for not being there in person. Maybe one other day, but happy to do it online anyway. So yeah, so I'll today what I'll try to do is essentially go over um, some of the activities here in my program and some of the work that we do from small towers all the way to tall towers, similar to the ones that Tim operates down there. So yeah, I just kind of always throw this slide here because my students, they tend to get a kick out of it. So I'm from Brazil, from the Southeast. Then I went to A&M to do my master's and PhD, did a little bit of a postdoc before at North Carolina before moving here. And I tell them that if I move again, I'll probably end up in Churchill or in the Hudson Bay. So probably good for now, and some of those dangers of extrapolation, I suppose. Um, so essentially, there are two projects that I'm involved with, essentially kind of in this broader theme of micromet measurements in agriculture, meteorological measurements in agriculture, and um, I'll talk about components of those. So the first one is essentially our eddy covariance projects here, our network, where essentially my role is um, carbon and water dynamics, right, in different cropping systems that we have here in the prairies, right? So this project is called LEAP, and I'll go into a little bit of more detail in the next few slides, right? Some of our funders, supporters here. And essentially what I'll discuss today is the work that my master students are doing. So one of them is Emily, the other is Julia, and they are essentially looking at things like comparison between a conventional business as usual type of a cropping system to one that adds cover crops. So things like intercropping, red clover in canola. So that's what Emily is looking at. And Julia, she's essentially looking at another comparison, but on the perennial side of things, and particularly looking at when perennials are terminated, what happens during this before and after the termination, right? And then the last piece to this is something that we recently started. It's a different project. It's called Canon to All Net. So led by the University of Guelph. And my role in that project is to do some of the tall tower measurements similar to what team has been running for the past number of years there, right? So that's more or less what I will discuss. Um, so when it comes to this first eddy covariance project, just a little bit of context here. So the name of the project, the acronym is LEAP, but what it stands for is leveraging ecosystems to transfer agriculture on the prairies. And the idea of this project is essentially to look at how land is being managed, right, in agriculture, and start to quantify some of the important parameters towards net zero, towards sustainability and profitability. Now, here there is a map of our sort of main soil order, the Chernozems, so grassland soils. In Manitoba here, we are particularly in the black soil zone, right? So part of the name of the talk. And when it comes to this other map here, kind of zooming in a little bit between Manitoba and Saskatchewan. So one of the components of this project is what we refer to as a landscape analysis, right? So essentially, go out there and sample what the farmers are doing, essentially, right? So, and at a different, let's say, level of intensity, if you will. So anything from soil samples once a year to maybe some biomass collection, things like that, all the way to where I tend to be what it's called. I guess we're in a Olympics kind of a frame of mind where we assign this sort of tiers of sites in terms of gold, silver, and bronze. And the gold sites is where we have some of the eddy covariance instrumentation, and that's where I'm involved. But essentially, the idea is to look at what we are referring to incorporating perennial function in cropping systems, right? So if we think about how land is typically managed in agriculture, right, we have sort of our business as usual, conventional annual crop rotation, which for us here is typically wheat, canola, oats, some others in the mix, fairly long rotations before repeating the same crop. Then on the other end of the perennial gradient, if you will, we have our obviously our perennials and forages, right? We're essentially permanent land cover for a number of years. And then we have sort of the in-between where that could be cover crops, intercropping, grain forage, rotations, things like that. So essentially, that's kind of where our goal is to sort of understand how those practices are essentially moving the needle, right, towards those goals here. Now, I guess I should say the landscape activity is just one of many. So there is a modeling component, there is sort of a social sciences component to this, where the farmers that are participating, they also are interviewed to kind of get a sense of what are the motivations and the drivers for their decisions on how to make certain management choices. So it's very encompassing, and obviously my role here is just on the Micromet measurements. Um, so I guess zooming in even further, so when we were kind of at the designing stage here, one aspect that was important is soil type. So here, what I have is sort of where we are here on campus at the university and some of our sites. And on this other side here, we have sort of a surface texture map for the province. And as you know, soil type influences sort of management decisions and also how good or bad they turn out to be, right? So getting the soil the soil texture as a factor in the study was really important. So if you see here, what we try to do is within this idea of business as usual, fully perennial, and somewhere in between, we also try to match that with sort of a similar systems in both a sandy soil cluster and a kind of a more fine textured cluster. And what you see here is a little bit of the pictures here, right? And you see here corresponding to our surface texture right there. So we try to replicate essentially uh, the same idea on both soil types, right? And because we know, for example, when it comes to cover crop, some growers in the region, they tend to be a little bit concerned with water use and potentially depleting plant available water. So that is a concern on this area. Flooding and waterlogging is a concern on sort of the clay soil types. So again, there is sort of there is a soil texture or soil type component there that kind of is important to take into consideration, and we try to look into that during the planning phase of the project. So then here, essentially, so what we are doing since last year, so we started a network of 6 eddy covariance towers, and essentially measuring, carbon water fluxes, some other biomat variables, things like NDVI, transmitted PPFD, soil water content profiles, temperature profiles, surface temperature. So all those sites, they are essentially identical in terms of instrumentation. So we're replicating all the same variables in all of those sites. And when it comes to sort of our clay sites or clay cluster, if you will, so we do have essentially our kind of a conventional business as usual convention, which 2025 was essentially just canola. Then we do have our perennial, which is just an alfalfa field that harvested for hay, 2 to 3 cuts a year. And then this particular grower, so these are all commercial farms, commercial operations that we are working with, right? Both clusters. And then this particular grower here, his strategy is essentially to intercrop the red clover with the canola. So that essentially, once the canola is out, red clover essentially starts growing immediately, right? So that is one of the topics that my graduate students are working. So I'll present some of the preliminary data that we have for this particular comparison, annual rotation versus annual with the intercropped red clover. And then on the sandy sites, then it's sort of a similar idea, but then here, it is the perennial in rotation with cereals or corn. So I'll present here what my other graduate student is working on in this comparison. And then here is essentially the one that was terminated this fall, and it's going to follow with 1 year of corn and then go back into perennial. So a comparison there. Um, so kind of I guess what this is a slide about what I am actually interested in when it comes to agriculture. So uh when I was thinking about this, so we are in a semi-arid area. The prairies technically are classified as semi-arid. And here we kind of have this pendulum that keeps swinging between flooding and excess water. But collectively, 70%, about 70% of the insurance claims, they kind of split even almost between flooding and drought. So we're always every few years of drought followed by a big flood and sort of in this situation. So when I during the planning phase, when we were thinking about this, particularly for the drought aspect of this, and also attaching to the soil texture component, the question that I'm interested is, okay, so when we think about the potential that we have during the frost-free season to essentially sequester carbon, right? So how much of that potential is reduced just because of soil water limitations, right? And within some of those different practices, right? Which ones are the ones that make the most efficient use of the soil water during the frost-free season, right? So essentially, a simple hypothesis there that if we can have maximize the soil water during this period, that's where those are the practices that should win in the comparison, right? But kind of conceptually, what I'm thinking, and this is not necessarily a new idea, but so typically most people would interpret this as the water use efficiency kind of diagram, right? Where you have your cumulative ET and then your cumulative net ecosystem exchange productivity. Then here, what I'm doing is minus the harvest removals, but instead of looking at the slope, of this line, which would be typically your water use efficiency. I'm interested in if, essentially, because I don't know if I'll find it once we start populating this plot with dots, if there is a break-even point, meaning is there a cumulative ET that after which we account for harvest removals, that's when we start seeing carbon gains, right? So a little bit of a leftover of carbon in the field. So that's kind of my sort of long-term vision for this is to see how much, essentially going back to this question is, okay, after we account for harvest removals, how much water do we need to have to essentially try to move the needle past this point right here? So that's kind of and I don't know if this will happen, but that was sort of my idea for the work that we are just beginning now. So so I'll start with the sort of annual versus annual plus intercropped red clover. And what I'm showing you here is just kind of the general 2025 growing season for us. And there are a few aspects that I think are important, particularly for the growth of this intercropped red clover. So solar radiation, pretty similar between the two sites, as we would expect. They are fairly close to each other. Air temperature, fairly similar as well. And one of the things that we noticed that there was, for the most part, cumulative rainfall was similar. We had one big event right here. They had an extra 30 millimeters of water, so we see that being reflected in the soil water content progression here. But other than that, somewhat similar cumulative rainfalls. But the first aspect that I would like you to keep in mind as we kind of progress on this part of the presentation is last year, so if you follow here the air temperature line, our air temperatures, they actually became negative, okay, well into November. So this is going to be really important for the growth of that red clover, essentially moving forward. So our first frost was essentially well into November, essentially. Um, and the other aspect here is the farmer that we're working with, he leaves a good amount of residue. So if you compare here, kind of our conventional with the sort of annual plus cover crop, and this is somewhat reflected here in the water content, right? So this is at 5 centimeters, so surface water content. And we see here that generally there is more water close to the surface. Um, so our interpretation here is just the residue suppressing a little bit of the evaporation, direct soil evaporation, and that will have some other consequences for this system right here where we are, particularly as it relates to soil temperature. So uh, I'm not going to go through all the eddy covariance stuff. I'm just going to go straight into the data and the carbon partitioning that is typically done. But essentially here, what I'm showing is the gross ecosystem productivity. So that is a fairly routine calculation based on eddy flux data. And the idea here is our measurements are showing exactly what we were hoping that they would show, that during the growing season, when both fields were just canola, they were essentially very similar. And where we see the effect of the red clover intercropped really comes after essentially canola was removed from the field, right? So around here, it took a while, but essentially then the red clover essentially um, started photosynthesizing and well into essentially November. So part of that air temperature data that I kind of referred to a little bit in the previous slide. Now, the other aspect here is in the bottom is the cumulative. And the first aspect here that I would like to point out is that in terms of the photosynthetic activity of those fields, so the red clover did increase. So we see that there is sort of the inflection point here after the red clover starts to essentially dominate the field, right, and grow. So there was essentially the effect that we're hoping for. But one other aspect, which kind of goes back to the increased soil water content, some more residue, we see here that early on, uh, the field that had canola and more residue, it had a somewhat lower GEP early on, and then essentially became the same. And this is reflected a little bit on the cumulatives right here. So the measurements started 1 week from each other apart, but you see that essentially in terms of the top here, there was a little bit of a lag, right? That we think it's related to that essentially too much water, maybe the soils being a little bit too cold, that kind of halted the development a little bit early on. But again, the cover crops compensated for that later. Now, when we move to essentially respiration, uh, we see also what we were hoping to see, that both fields behave exactly the same in terms of respiration. But what we saw in the cumulatives essentially uh, very similar to each other. But then after the canola was removed on the what we're calling the diversified field, we do see an increase in respiration. And this obviously is associated with the growth of the red clover, some of the residue at the surface that is also respiring. The temperatures are warm here, right? Um, so we do see an increase on this due to the presence of the cover crop. And again, we do see again this sort of inflection point in terms of respiration increasing past of the just the conventional field, right? Um, so when it's all said and done, so I guess in preparation for this figure, what we're seeing from this first year of data is that in terms of the cumulative and kind of the daily progression, so early in the season, due to what we think is too much water, maybe a little bit too much residue, maybe the temperature is getting a little bit too cold, we do see that there is sort of a little bit of a lower net NEP for the canola plus cover crop, right? It catches up later, but we clearly see that there is an effect there in terms of how much it peaks just on the initial phase and then essentially becomes the same. But the important piece to this, and which is relevant for us here, as in Minnesota, the shoulder season are pretty short, right? So last year, for all purposes, it was pretty unusual in the sense that we essentially had our first frost in November. So prime conditions for shoulder season growth, especially because there was also water. And what we see here is that after canola was removed from the field, we had about 60 to 70 potential days where temperature was essentially higher than zero. And what we saw is that out of those essentially 60 to 70 days, it took about 40 days, 38 days for the red clover to essentially overpower the respiratory losses from the background, from the canola that was removed. So out of the 60 to 70 days, potential days that we may get, there was only carbon fixation or net carbon fixation in only 29. Okay, so I think that is pretty relevant for us here. So it did move the needle. So that initial loss, or that we are attributing, at least for now, to soil water, maybe lower temperatures there, um, it did essentially kind of level the gain here for both systems. But for us, the I think the relevant result here is that however long this shoulder season is, so it looks like we're gonna need at least some 30 days for it to move from a source to a sink, and then it will remain essentially as a sink for probably 30 days here. It's probably going to be the maximum. Who knows? As temperatures are increasing and changing, this could be stretched a little bit more. I don't know how much more, but this is kind of our interpretation there, and I think the relevant result here. So this is the evapotranspiration from both fields. And essentially here, sort of a kind of following the same trend as during the growing season, both fields essentially being the same, but early on, the canola field that had no, our business as usual, very little residue, mostly bare soil exposed, higher ET early on, and the other system a little bit less. And even after the canola grew, or sorry, the red clover grew, it didn't really fully had that same inflection point that we saw in the carbon. So it remained low, especially because um, not as much residue is left in the field. So the water use rates, again, uh, cumulatives, somewhat similar, but showing some differences there that we probably can expect based on how the systems are run. And then here, I think it's sort of the interesting going back to that kind of water use efficiency idea. So the during the growing season, the canola fields they were the same as we were hoping that they would be, but then the cover crop effect or the intercropping of red clover coming after is really showing here, right? So we do see that this increased water use efficiency in the shoulder season essentially moves the needle for water use efficiency, the comparison. So 1.9 for the canola plus red clover versus 1.6 here on average. So it did move the needle a little bit. So obviously we are very early in this. We are still not accounting for harvest removals and things like that. We're in the data collection process, but that is something that we're going to see. That's kind of the next steps here is to start fully accounting for the carbon balance. But we're seeing some I think interesting results for our region here in terms of the carbon and water dynamics of those systems. And that's what Emily is going to have her thesis on. Uh, so this is the other study, the perennial. So that's kind of a here of time lapse progression. So I'm going to switch to the other part of the project, kind of the lower um, cluster where we have our sandy soils. And essentially here, so we had in 2025 for the conventional business as usual spring wheat, right? In 2026 is going to be canola. And for the perennial annual rotation, so 2025 alfalfa, and then it was desiccated in the fall and essentially disc tilled. And next year is going to be corn, and then it will go back to perennial. So we're trying to catch this transition and what it does to the carbon and water budgets here. And you can see here sort of Julia right there, the other students, Nathan, Shayla, some of the students that work with me, kind of very working hard there in the field. Um, and what I'm going to do here is kind of go through the same kind of data sets showing the differences between the two systems. And what we're seeing here, so on this top panel, that would be our perennial or alfalfa, and the annual here is spring wheat right there, and then the cumulatives. And essentially, in terms of the photosynthetic activity for the for both systems, it's doing what we expect it to do. So following the phenology for spring wheat, right? And then after a cut, photosynthetic activity going down, then going up again, then another cut, then going up again, then another cut, and then essentially termination here with the herbicide application, and then essentially kind of tapering this down. What we are noticing here is that this recovery period for the photosynthetic activity of the field to kind of go back to pre-cut levels for our conditions here is taking about at least 1 week, but most likely anywhere between 10 to 15 days for it to recover, essentially to pre-cut levels, right? So it seems to be pretty consistent. And we can see here sort of on the cumulatives that in the shoulder season, obviously, as we would expect, greater photosynthetic activity, but then the annual takes over. Then we have those cuts that are really slow down, right? But then eventually catches up um, on the um, on the shoulder season again. But in our case, because of the termination, they were essentially very similar, right? So it was not, and we were expecting that to happen, but not as high as essentially could have been just because of the termination. And here is where I think we see the real effect on the ecosystem respiration. So essentially here, in terms of respiration rates, for the annual field following the sort of the phenology of the crop and then kind of a slowing down. And we see this big effect here, and we see roughly around here. So here's the desiccation, and then the disking actually happens around there, and just a much bigger respiratory loss just because of those activities. And then this is essentially reflected in the cumulatives, right? So a pretty substantial difference in terms of the total. So 260 grams of carbon per meter squared when you account for the losses due to termination, right? So again, uh, one of the things that we were trying to understand is there's some data from termination in clay soils, but in sort of sandy loam soils, we don't know the size or the magnitude of those termination losses. I should mention here that the farmer did apply manure in the fall in a split application. So half of it was in the fall, the other half is going to be sometime this spring. So we are not accounting for that, but that is going to be part of the carbon budget essentially. And then once essentially all is said and done in terms of net ecosystem productivity, so essentially doing what we were hoping that it would do. So after the cut, NEP becoming negative for the perennial, anywhere between 10 to 15 days to recover, then another cut, then another 10 to 15 days to recover here. And then what we see here is essentially, so around day 67, so there was termination right here. And then disking in this part right here. So essentially this big loss here, okay, in the shoulder season. While in the perennial, there were losses after harvest, but then with just weed and volunteer growth, it essentially became neutral, right? In this period here in the shoulder season. So in terms of cumulatives, it essentially flattened out because of this kind of neutrality here. While in the other one, it essentially went to almost zero. So a difference here that is essentially a factor of 100, right? So again, in these fields, we are not accounting for harvest removals just yet. Um, and the manure application in the perennial, those are going to be part of the carbon budget calculations. But this is a pretty substantial loss here in this particular soil type. Then this other piece is going to be the ET, and again, ET slightly higher during the shoulder seasons for the perennial, following the same pattern of going down when the cuts were happening, and then essentially after termination, essentially going down. And we see some ET here from soil, direct soil evaporation and um, uh, the volunteer growth and weed growth. But in terms of annuals, they're somewhat similar, but we higher ET for the perennial here, mostly due to those peaks here between the the main growing season. And then lastly here, that's kind of the water use efficiency, which is a reflection of what the management is doing with the annual peaking during the growing season, perennials peaking in the shoulders, but then with the termination, right, then we see essentially the water use efficiency as we would expect, essentially plummeting below that of the annual. Um, so again, the termination there having a really strong influence on the water and carbon dynamics of those fields. And um, for this year, what we are excited is to see is corn. Obviously, it's a very productive plant. So, how will that change essentially from this big respiratory background because of tilling that we currently have on the perennial that was terminated? So, how long it will take to become positive again, and then once you go back into perennial, or how long it takes. So, those are the kinds of questions that we're interested in quantifying because we think it's relevant for management, right? So, uh, the last piece that I want to kind of refer to is this project called Canon to ONET that I alluded to, which is led out of the University of Guelph with Claudia Wagner-Riddle. And what I'm showing you here is just kind of a snapshot of what the project is. So essentially, we have uh, uh, flux gradient sites, so equipped with trace gas analyzer, so a TGA, a laser analyzer to measure N2O when measuring gradients. So using the flux gradient technique. So in Ontario, we have 3 sites, 2 sites in Manitoba, 1 site in Saskatchewan, and 2 new sites in Alberta. And part of this project, we established the tall tower, and that's where I'm involved with. And within those sites, lots of activity in addition to the Micromet measurements. So looking at essentially soil nitrogen, some microbial work as well. I cannot speak too much about this because we have our experts in the project essentially leading that, but they're doing also tracers as well. And my role here is with the tall tower. And Tim has given us lots of guidance and advice on how to do this. This is kind of a picture of our lab setup. So when we're putting everything together, so essentially we have the same analyzer as Tim does, the Picaro N2O carbon monoxide analyzer. And we have for CO2, the Lycor 7000 analyzer. So essentially, getting N2O and CO2 from the tall tower. And what you're seeing here is kind of a picture of a lab test, but what we're trying to do is try to get a test response on the, because the intakes at the tall tower are around 100 meters. So just a pulse test. So we're pulsing a high concentration CO2 at the intake and essentially looking at how long it took after the pulse after the pulse was injected here so that the analyzer could see. And for the CO2 analyzer, what we found is that after the pulse for the peak response, it took about 45 seconds to travel 100 meters of tubing plus going through the analyzers. So this, so 100, so 45 seconds, but this full cycle, so from the poles to the peak until it kind of tapers down again, is about 2 minutes. So, which I think it's pretty good resolution to be able to see an event in about, a minute or so. I think that's pretty neat. So, something that we're learning how to do, and very thankful to Tim for sharing his expertise with us. This is a sort of a picture there now of our Tall Tower site. So this is in the province of Saskatchewan, the city of Yorkton. It's in a broadcast tower as well. Picture of our intakes here. Then you can see the potholes there in the area. And essentially that's our system there when we first deployed it and with the CBC tower manager there, kind of essentially making sure that we were not doing anything wrong during the installation. And so part of this work that we essentially started in December last year to now start looking at some of the top-down constraints on emission models and things like that for our region. And yeah, so I kind of put this picture here because folks tend to get where are your intakes? So I said, oh, looks like you need to go to the doctor, right? So, but ours are right here, just below the strobe lights in the top tower. So barely can be seen if you're just standing there, but zooming in a little bit, then we can see our intakes. So just for perspective. So a couple of things here. So we've been sort of monitoring this and also um, in the phase of developing our data processing scripts and things like that. And what we saw here, so in February, and I'm assuming you all also had some pretty warm days. Um, but what happened here was we did see a pulse here. So our uncorrected values for N2O, so there is a little bit of an offset that we need to correct using our calibration tanks. But we were around 347 uncorrected values, ppb. And then we saw this pulse here going up to 356. So essentially increasing by a factor of almost 10 parts per billion. So it's okay, we had maybe a small thawing event in February there. So and more recently, so this is a Teams, he very graciously ran the footprint for us. So we could get an idea, at least for our tower, using a few weeks of data. So this is thanks to Tim. But this is our data. And again, this is really rough, not very, so lots of quality control still needed to be done. But what I wanted to highlight here, so this is data from January all the way to essentially end of April. And this is N2O on the Y, and then time on the X. And we saw this 2 weeks ago, this really absurd pulse of N2O at our site. So it actually went above, so this is uncorrected N2O, but actually went above our calibration tank here that is 380. So we saw this massive, massive spike. And we are kind of working towards cleaning this up a little bit. So we can see if this makes sense, and it would be nice to actually compare this with Teams data as a to see if you all saw the same thing at your towers, and also as a sanity check for us to make sure that this is not a problem with the analyzer in an actual response, but because it kind of went back to sort of our baseline ambient, seems like this is true, at least at a first glance. So yeah, that's sort of what I have for today. I'd like to thank Tim and John as well for the invitation. This is the already mentioned funders, colleagues, a bunch of people that are helping with this project. And here's just a couple pictures there of me and my students in the field, and the Micromet Christmas tree, Tim. So that is a nice touch there by the students. I think they think the Micromet lab needs some decoration. And I'll have any take happy to take any questions.
Okay, thanks so much, Enrique. Questions for the audience? Thank you. Let's see if there's any. I'll check the chat here. Oh, looks like Dave Mulla is he raising his hand?
Yeah, I can ask a question.
Yeah, thanks, Dave.
Um, so the cutting of the alfalfa was a very interesting result. It really reduced the um, the net ecosystem productivity compared to the annual system. Um, did you see any effects like interactions of that cutting on water balance as well? Uh, ET, for example, and how that might have also, I mean, is the effect on net ecosystem productivity just because of the change in biomass, or is it also due to feedback loops with soil moisture?
Thank you. That's a great question. Yeah. So what we saw in terms of the relationship with so on the photosynthetic activity, so essentially it's a canopy loss, right? So you're losing green leaves that are photosynthesized, so this going down. Um, but on the ET side of things, so we do see the same reduction from pre-cut, right? So around close to 6 millimeters here, pre-cut, then going down. It doesn't go to zero, which tells us there is some soil water evaporation, right? Then it kind of goes back up. Then there's another cut right here. So baseline here, I want to say post-cut, what it looks like 2 millimeters. Then it goes back up again, and there was another cut here. So I think there is, as you mentioned, a component of direct soil water evaporation after the cut that maintains that AET. In terms of the cumulative here, they turn out to be pretty similar in terms of so a little over 400 millimeters for both fields. And what we are essentially attributing this is the fact that the perennial had slightly higher cumulative is this period here after, right? So the annual was harvested. So here essentially going into senescence and higher ET here where this canopy is essentially yellowing and then this is still active. So we see that a bit of an inflection point here, but then after the desiccation comes in and essentially termination, there is some soil direct soil water evaporation, but then it tends to flatten out, and so cumulatively, it was a little bit higher, but not by much. But there is sort of and then I'm showing here the water use efficiency for comparing the two systems. So yeah, to answer your question, yes. So I do think there is a um, uh, uh, soil evaporation component here after the cuts when this water use efficiency here goes down. Um, it's just we have not made any attempts to try to partition ET in the perennial field just yet.
Enrique, can I just follow up on David's question a little bit? What were some of the factors for terminating the perennial system? I think you
Mhm.
mentioned, um,
Yeah.
did you need, could that have been done later to sort of retain more carbon, or what were the factors there?
So that is a common practice for some of the dairy and mixed growers in the prairies. They tend to terminate after, uh, they start seeing some declines in the productivity of those perennials. So it's something that they do to renew, renovate the system. So that's kind of what we're trying to capture. So they try to sort of rotate to have the benefits of essentially the perennial plus a rotation with an annual and then go back into the perennial. So that's kind of it's part of a management strategy that is very common here. And the idea is to look at and see, okay, what is that termination and then the effect of, let's say next year is going to be corn, but we have another one that is oats. So the post termination does the type of crop influences that. And then after you go back to the perennial again, how long would that take? So that those are some of the questions that
Yeah.
we are working towards. We're very early on, so I wish I had the full data to show those results.
Okay, thank you. Other questions for Enrique?
Just following up on looking at this figure and the difference between the annual and the perennial there. I know you showed precipitation on the last one, but is there any sense of whether the water use efficiency, the water needs are lining up better with the natural precipitation patterns in one system versus another?
Yeah, that's a good question. Yeah, so the precipitation was very similar for these fields as I showed in kind of that previous slide. Um, so what we are seeing is that um, it is more about soil water uh, after the cut. So it seems like if you cut it and then there is a good amount of soil water storage, maybe that post-cut growth can be a little bit faster as opposed if the soil is too dry. Then it can stretch a little bit more than that 2 week period, roughly. So the precipitation, so to your point, I think yes, it's important, but I think it is sort of a combination of soil water plus um, when the precip happened and if it was enough to recharge during that regrowth. So that's my, at least for now, my interpretation.
Yeah, I'm interested in that. I was kind of eyeballing in your first paired sites. It looked like the one with more perennials it had more soil water storage throughout the year, but it's like the water it collected during a rainstorm seemed to stick around more. And
Hmm.
that's something I'm really interested in on the soil health side in terms of soil structure. Like, can you interpolate from this data that you're essentially capturing and retaining more water that falls from the sky because of your soil properties? So I'm always interested if you can take it into the soil side.
Oh no, absolutely, and that is something that we are hoping to do this year, to because we're measuring soil water content profiles in all of the sites. So we measure down to 60 centimeters with the dielectric probes. So we plan to incorporate that in the data set, so a calculation of soil water storage. And what we're hoping to do as well this year is to run soil water retention, hydraulic conductivity, to start looking at if there are some of those changes that you are referring to. Um, that can potentially be influencing the water storage component of this. So yeah, we're thinking about it, and we're in the process of learning how to run the high prop and the KSAT and doing those kinds of measurements. But we're hoping to tie all of this together.
Awesome. Thank you.
Thanks, Anna. Any other questions? Oh, Carl has his hand up. Thanks, Carl.
Yeah, hi Tim. Thanks for the seminar. Very interesting. I was interested in the inhibitor part. Have you gotten any data with that, or what's your
Um,
thoughts on that? And what kind of inhibitor were you working with?
yeah. So we do have some of the the inhibitors, they're being tested on the flux gradient sites. The 2025 data is being processed right now. I cannot speak much about it because we're still kind of working through the data sets, but they're being tested. And what I can tell you is, as you can see here, essentially the idea is try to match with the practices across the flux gradient sites, they're kind of similar, but also tailored towards the region. So, for example, Eastern Canada, more humid, more rainfall, and then as you go into the prairies, then you start getting into rain shadow of the Rockies, right? So, essentially a drier environment. Um, so here, the inhibitors are kind of being coupled with irrigation. So that's kind of what
Mm.
they are studying at in Alberta is trying to see kind of that interaction right there. Um, um, Here in Ontario with Claudia, they were looking at the interaction with cover crops and things like that. I'm sorry, I cannot speak too much about the results just yet. But what I can say is the inhibitors are trying to they're being used in a way that makes sense with the management in the different provinces, essentially.
So are they applying it with fertilizer then, or just playing? So I assume that's what they're doing. They're applying
Yeah,
fertilizer with the cover crop and using
yeah,
an inhibitor
yeah,
with. Okay.
yeah,
All right. Well, good. I'll
yeah,
be interested to learn what you find out.
yeah.
Interesting.
So year essentially year two of Canon 2.0 Natch. Um, and again as well trying to kind of coordinate this big ship
Yeah.
that is this project. And I do think that one of the outcomes that's going to be very interesting here, what we're hoping to see is that how these different options, right, in terms of nitrogen management, how do they vary with climate, right? Because
Mhm.
we're going for essentially really semi-arid in Alberta to essentially very humid conditions in Ontario, southern Ontario. So what is the kind of the interaction there? So
Great. Well, thank you.
thank you for the question. Yeah.
Thanks, Carl. Any other questions? Enrique, can you go to your tall tower slide,
Yes.
the data point?
This one here.
The high
This
concentration.
yeah, this
Yeah.
one here.
So that's interesting. So I guess just wondering if you also saw carbon monoxide also be high on that particular event.
Tim, good question. I'll have to go back and look at it. This is something that we have not plotted yet,
Okay.
but we'll start looking because are you thinking about tractor activity?
What I'm thinking about is, in many cases, if you see really extreme values in both signals, it might
Mm.
indicate an instrument problem. So,
Okay.
but also, CO acts as a tracer of um, fossil-based N2O.
Mm-hmm. Mm-hmm.
And so, based on where you're located, I would be surprised if that was the driver, given the environmental conditions. This sort of um, at your latitude beginning to warm up and see some high temperatures. So I'd just be
Mm-hmm.
curious if the CO, our background CO here in St. Paul, typically around 130 parts per billion, 120 parts per billion.
Mm-hmm.
It's pretty steady, but we see a little bit of an urban influence,
Mm-hmm.
so wind direction influence. We were hoping to sort of use that to partition out ag N2O versus
Mm,
fossil-based N2O. So that could be one diagnostic that you could, use to help convince yourself that this is a real event.
okay.
Yeah, so something to keep an eye on. Of course, you have CO2 as well to take a look at.
Hmm.
Um, but I'm pretty sure that some of our winter or our early spring freeze thaw stuff, we can see up to 370 parts per billion. So
Oh, wow.
you're
Okay.
you're seeing 40 parts per billion greater than the background N2O concentration,
Hmm.
um, which is, there's no very little high frequency data in which to see this and to look at it simultaneously across sites. So this is pretty exciting.
Yeah, yeah, I thank you, Tim, for the comment about CO. We'll definitely look into it and see if we can find some synchronicity there.
Yeah.
Yeah, and I appreciate, yeah, because for us, like I said, we're learning how to do this, so we welcome your input and your feedback because we're pretty new to the Tau Tower. The students call it Frank, as Frankenstein because they call it it's like a monster.
Okay. Well, thank you. Any final questions for Enrique? Okay, I'm not seeing any hands or anything in the chat. So I want to thank you again, and Enrique, and we look forward to working with you. You're relatively close, so next time when your visa stuff is worked out, we'll have you come down in person.
Absolutely. Looking forward to it.
Yeah. Thank you. Thank you for your time. Thank you, everyone.
Okay. So
Yeah.
I'll log out here.
Okay. Okay. So anyway, keep me posted on the data. This is very exciting.
Okay, sounds
Um,
good. Yeah.
and with a little bit of data, I should be able to we should be able to run a little mini inversion to see how things look.
Okay, sounds good.
Yeah.
And what I will do is Claudia's postdocs are kind of helping with the script side of things.
Okay.
Because I started one of script, but with teaching and stuff like that, I got slowed down. But have you met Patrick? He was Andy's graduate student, Patrick Powell.
I don't think I've met him in person, no. But
Okay.
if he's worked with Andy and he's been in working with Zorn and MATLAB and all that stuff, that would be super valuable.
Yeah. So and so Claudia has two postdocs that are very capable with instrumentation and data processing, Jason and Pat. So we're kind of so this is sort of something that happened early this week, this plot here where
Oh,
we're
okay.
kind of it's kind of fresh out of the
Yeah, no, no,
oven.
that's great. You got to
So
start somewhere. And all of this processing can take a lot of time. I've sort of reinvented some of my scripting and how I've done things over the years, and some things are just not as clean as I had hoped, and you get busy with other things. So it's always a work in progress.
absolutely, yeah. But we will definitely contact you as we kind of get a little bit better. I think
Yeah.
we're too crude just yet.
Okay. All right.
All
Well,
right.
keep it going. Yeah, that's exciting. So thank you again.
Yep.
All right,
Yeah,
Enrique.
absolutely.
Yeah.
Thank you,
Bye bye.
Tim.